everyone, my name is Namrata Singh and I've done my Masters in Animal Sciences and I've been teaching zoology from past 2 years. So today we'll be studying about excretion, mainly focusing on the structure of nephron and urine formation. First of all, what is excretion? So it is the collection, separation and elimination of liquid nitrogenous waste from our body. And since we are human beings, we produce urea as major nitrogenous waste. So how is that urea formed? It is formed in the liver, where the deamination of amino acid takes place, which produces ammonia, and that ammonia is converted into urea in the liver. And this urea containing blood is taken back to kidney. Now the blood which contains urea also contains many good components also, for example glucose, vitamins, minerals, which is needed by the body. Now what happens is that we need separation of the good and the bad. We need separation of what is useful for us and what is harmful for us. This separation is done with the help of nephron. So nephron are the structural and functional unit of the kidney. Now if you look at the structure of the kidney, so it is a bean-shaped organ, and this blood which has urea will be taken to kidney with the help of renal artery, and the filtered blood will be taken back to the heart and to the body with the help of renal vein. Now if you look at the structure of one kidney, then the outer part is known as cortex and the inner part is known as medulla. And the part near the hinge, this side, is known as renal pelvis. Okay, so a single kidney can have around 1 to 1.5 million nephrons. Alright, so if you look at the structure of the nephron, this cup-like structure is known as Bowman's capsule. After that, this Bowman's capsule, it opens into a highly convoluted structure which is known as proximal convoluted tubule. Proximal because it is nearer to Bowman's capsule, convoluted because it is highly coiled, and tubular because it has a tube-like structure. This PCT now opens into a loop-like structure known as loop of Henle. Okay, this loop of Henle also has two arms. The one which is coming down is known as descending limb, and the one which is going up, which is a little bit broader also, as you can see, is known as ascending limb. After this broad tube also, it becomes narrower again, and this narrow tube again is known as distal convoluted tubule. Distal, why it is distal now? Because it is distant from Bowman's capsule. Convoluted because again highly coiled, and it is tubular in structure. This DCT now opens into a collecting duct, which is common for many different nephrons. Now if we look at how the separation takes place, it takes place by 3 methods. One is ultrafiltration, second is selective reabsorption, third is tubular secretion. This ultrafiltration takes place in Bowman's capsule. So if we zoom this Bowman's capsule, for example, this is Bowman's capsule, and now one arteriole will come inside it, taking the blood which is urea. It will take the blood inside it, and this is known as efferent arteriole. It takes the blood inside this Bowman's capsule, and then this arteriole, it divides into many capillaries. This bunch of capillaries is known as glomerulus. Okay, and then again these capillaries, they join back to form efferent arteriole. As you can see, the efferent arteriole, the diameter is thicker, and in the efferent arteriole the diameter is thinner. What happens? This creates a high pressure in the glomerulus, which helps in the ultrafiltration. So the filtration which takes place in high pressure is known as ultrafiltration. And also this visceral layer of Bowman's capsule, it has many pores, and the cells are podocytes. So this pores, they helps the filtrate to come in, and now this filtrate is taken to the PCT. Okay, so now in the PCT, actual selective reabsorption takes place. So now ultrafiltration, it happens in the Bowman's capsule, and the selective reabsorption now happens in the rest of the tubular structure. So in the PCT, mainly you can say the glucose and amino acid and some amount of water and your sodium and potassium. These are the things that are absorbed back to the blood, and most of the processes, most of the components that are reabsorbed takes place with the help of active reabsorption. Okay, so energy is required there. Then in the coming descending limb, so it is porous only to water. Okay, so only water is absorbed here. It is impermeable to any kind of electrolyte present in it. Now coming to the ascending limb, so it is almost impermeable, you can say almost impermeable to water, so no water will be absorbed here. Only some amount of NaCl, some amount of bicarbonate, you can say, is absorbed over here. After that it comes into DCT, and in DCT again some electrolytes, for example your sodium and your again bicarbonate or NaCl, these are the things which will be reabsorbed. And in the collecting duct, mainly water is reabsorbed. Okay, most of the water is reabsorbed in the collecting duct, because here only your urine will become concentrated. After that we will come to tubular secretion. Now some of the components might escape this filtration, this ultrafiltration. So in that case, the blood which is flowing through all of these tubules will pass this component directly into the filtrate. So that is tubular secretion. It mainly takes place in the PCT and DCT area only, mainly in the PCT and DCT area. So in the PCT area, your excess amount of H ion will be given back into the nephron. Your uric acid will be given back. Then creatine will be secreted into the filtrate. And in the DCT area, again excess amount of H ions, your potassium ions will be given. Then some amount of HCO3 also can be given. Okay, And in the urea, in the collecting duct also, sometimes the urea is also secreted back to maintain the osmolarity of the urea formed. Okay, so this is how. And now after that, all of the things have happened. Now it will come into the collecting duct. Okay, so all of these collecting duct, they rejoin in the renal pelvis region, and after that, drop by drop, the urine will come into the urinary bladder with the help of ureter. And when your urinary bladder is full, you will get a sensation in your brain that you need to pee, and after that you can release the waste products from your body. So we studied the collection, separation and elimination of the waste. This waste is produced in the liver and it is taken to the kidney, and in the kidney, under filtration, selective reabsorption and tubular secretion takes place, and this is how the urine is formed and it is excreted from the body. Thank you.